Okay, today I will start after some time, getting some time to take class. So it will be cell theory. You have the first chapter is concepts in cell theory. So I'll share my slides. Is it visible?
Yes, sir.
Sir.
Okay. Okay. So what is the key concept of this cell theory? Can you see this hand moving?
Yes, sir.
Okay. So we are talking about this thing called cell theory. So what is the key concept? The key concept is cell is the basic unit of life. What do you mean by cell? What is cell theory? The gist is it is the basic unit of life. And this is a electron microscope picture. This is an amoeba. Can you see these little pseudopodia coming out from these amoeba? These little arm
Yes,
like,
sir.
these are pseudopodias. This is how pseudopodia looks like. You see, normally you see them in a textbook diagram, but this is how it looks like. These arms are coming out. This is a basic unit of life, one cell. Just 1 second, I'll allow one person to join. Okay. So you are able to still see them?
Sir.
Yes,
Okay.
sir.
So many scientists, they worked and they developed the theory of what a cell is. So many of them, they worked to one by one, not together. They developed on a concept. First one was Robert Hooke, Anton von Leeuwenhoek. They made the microscope. Then the real actual theory was given by Shildon and Swan. They were the main proponents of this theory, and it was even developed by Virchow. So normally you people have read about the Shildon and Swan cell theory in your early works, early class 11, 12 things, the cell theory by Shildon and Swan. Actually, the theory of the cell theory developed when as the microscope improved, the cell theory started to develop even more. The cell theory started to develop even more. More advanced microscopes came out. Advanced microscopes and better the cell theory started to develop. So in whole of biology, the theory of a cell, it is an unifying concept. That is, it unifies botany, zoology, microbiology. Botany, zoology, microbiology, even physiology. Because The nutshell is cell is the basic unit of life. Life science, biology studies with life, and the basic unit of life is cell. So whichever branch of biology you go, the concept of cell stands firm. Early studies in the microscope, it led to the cell theory. The cell theory has got three principles. The first one is all organisms is made up of cells. All organisms are made up of cells. This is the first theory. This you can see some cells under the microscope. This is one cell, made two cell, 3 cell, 4 cell, 5 cell. The second theory is all existing cells are produced by other living cells. This is the second cell theory. That is, this you can see, this was one cell here. It is now dividing into two cells. So one cell is basically dividing into two cells. So cells come from pre-existing cells. From pre-existing cells. This is the second theory. First one is all life is made up of cells. There is no living organism which does not have cell. And the third and the most important theory, it is the most basic unit of life. If you break a cell, you find lots of components. But these components, they are non-living. If you break up on a cell and the parts of the cells are non-living. But when you take the cell as together, it is the basic unit of cell, which is life. Basic unit of life. Prokaryotic cells, you know that cells are divided into eukaryotic cells and you have prokaryotic cells. So there are lots of differences which you need to study. And you have studied a lot. Can anybody of you say give one difference between a eukaryotic cell and a prokaryotic cell?
Nucleus.
They lack a nucleus. And next, what else do they lack?
Membrane bound organelles.
Membrane bound organelles. Very good. So naam ki ke bolche?
Amrita.
Amrita. Very good. So
Sir, ribosomes.
Ribosomes. Very good. Eta ke bolle?
Oncon.
Oncon bhomik. So all cells share certain characteristics. All the cells. Prokaryotic cells, they lack nucleus and most internal structures of eukaryotic cells. So basically, some differences between eukaryotes and prokaryotes, even though you were a COVID batch. That's very good. One characteristic which they share is they tend to be microscopic. The cells tend to be microscopic. That means you see there, you can say observable under microscope. Under cells are observable under microscope. This is a electron scanning electron SEM. Whenever you see this word SEM, it means scanning electron micrograph. That is a image taken from a scanning electron microscope. So instead of light, normally in a microscope you use light, but in an electron microscope you use electrons. You give a beam of electrons. These electrons, they go and hit the surface. And if you have a structure, this is a flat plane. On top of this is one cell. The nucleus is here. Let's see who has come. So, দেখা যাচ্ছে এখনো ছবিটা? হ্যাঁ কি না বলো কেউ?
Yes, sir.
Yes, sir.
Okay, this is a cell on a flat surface. This is a cell. Now, you have an electron beam coming over here, and they get scattered in different directions. And these are captured on an image. So what you get actually, because by watching the scattering, you can get this, you can understand at the surface, how rough the surface is, the ups and downs of these curvatures on the surface. You can see it is scattered. So you get an image over here. So all these are scattered. You see these little details. That is because of the scattering. And this type of electron microscopy is known as scanning electron microscopy. This is 8,800 times. This particular cell has been magnified. It is 8,000. What you see is 8. This is 8,800 times much more bigger. Then the next characteristic is all cells are enclosed by a membrane, and that is known as the cell membrane. Some people may, in many places, they may call it a plasma membrane. You possibly have already, the class of plasma membrane has been taken. So all cells, characteristic of all cell is that they are covered by cell membrane. Then the next thing is all cells are filled with cytoplasm. All cells are filled with cytoplasm. They're inside the cell membrane bound thing. There is a fluid, semi-solid structure enclosure, and that is jelly-like cytoplasm. And you know there are two types of cells: eukaryotic cells and prokaryotic cells. As you know, eukaryotic cells have got a nucleus. This is a nucleus, a dart structure inside the whole of the cell. This is the nucleus inside an eukaryote. And you know that prokaryotic cells, they do not have membrane bound organelles. Prokaryotic cells do not have a nucleus. A nucleus is also a membrane-bound organelle. Nucleus is also membrane-bound. Membrane-bound organelle. Now you can clearly see this is a eukaryotic cell. And this is a prokaryotic cell. Prokaryotic cells do not have a nucleus. Prokaryotic cells do not have membrane-bound organelles. Now, another concept of the cell theory, the modern, now according to the modern, use the first 3 points hold. And this is eukaryotic cells share many similarities. All the cells, they have an internal structure. You can see that these little green lines, red lines, they are known as cytoskeleton. They can be actin, myosin, vimentin. So you have microfilaments, you have lot of nanofilaments, you have microfibers, etc. You have lots of they give support and structure to the cell. This blue colored, of course, is the nucleus. This is the nucleus. So this is the cytoskeleton has many functions. Like I said, it supports and shapes the cell. Helps position and transport organelles. Myosin filaments on these cells, you have some molecular motors. Motors which move. So you have this microfilament on which you have a motors, molecular motors. It's I'm drawing it like a car. They move on top of these filaments. On their head, they can carry low cargo. And what are these cargoes? The cargo can be chromosomes. It can be organelles. It can be molecules. They transport. So these filaments, they act like a road on top of which molecular motors move. Example of molecular motor is there are kinesin, dynein. These are examples of molecular motors. So when if you have read about mitosis, meiosis, there is movement of chromosomes. These chromosomes move by the help of these molecular motors on top of spindle fibers. A spindle fiber is also a cytoskeleton. It's a microfilament. They provide strength. হাড়ের মতন কাজ করে, like your skeleton. Like I said, during cell division, the movement of the chromosomes, this cytoskeleton helps. On spindle fibers. Movement of chromosomes. Like I said, they aid in cell movement. It helps the cell to move. Changes in the shape of these microfilaments in these areas. This change in shape. These changes in shape for movement. So they aid in cell movement. Microfilaments aid in cell movements. Now, the next thing is that several organelles are involved in making and processing proteins. The nucleus, as you know, it stores the genetic information. Many processes occur in the endoplasmic reticulum, which we will be studying. So what are the endoplasmic reticulum? This is the nucleus. Here it is the nucleus. Just hold on, somebody is ringing the bell. So from the nucleus, there are little these finger-like projections. They are known as the ER, endoplasmic reticulum. On top of these are these dot-like structures known as ribosomes. They are the sites. The ER and the endoplasmic reticulum are the sites of protein synthesis and protein synthesis and processing. So ribosomes, protein synthesis takes place. And in the endoplasmic reticulum, protein processing, folding, there are the glycosylation and various other modifications takes place. Okay. So now we know that the nucleus stores genetic information. We know that many processes occur in the endoplasmic reticulum. What processes? Protein processing. Endoplasmic reticulum, we know there are two types of endoplasm. Can anybody of you tell what are the two different types of endoplasmic reticulum? This is the first type of endoplasmic reticulum. This is the second type of
Rough and smooth.
rough and smooth endoplasmic reticulum. So you have the rough, ER, and smooth ER. On top of this, you have these little button bead-like structures. They are the ribosomes. So on the rough endoplasmic reticulum looks like rough. They are laminar and flat, and here it's almost like a tube, and they don't have ribosomes. No ribosomes. Rough endoplasmic reticulum, smooth, and smooth endoplasmic reticulum. Ribosomes, they join the amino acids to form proteins. They join these amino acids to form the proteins. Amino acids like lysine, leucine, methionine, they are all joined together, and a linkage of all, say this is first amino acid, second amino acid, third amino acid, fourth amino acid, fifth amino acid, like this, several amino acids together they form a polypeptide or a protein. Many polypeptides also can make a protein. Then there are lots of vesicles present. They are membrane-bound sacs that hold materials. You see these, there are vesicles over here. You see these vesicles? Are you able to see these vesicles? Blue colored? Yes, no?
Yes, sir.
Yes, sir.
Okay. So they hold materials. They can be little protein molecules or whatever, enzymes, whatever it is. They hold them. They are covered, they are enclosed in membranes. You have all heard about mitochondria. They look like this. They are basically essential important in making ATP. What are the processes which make ATP? One first is the citric acid cycle. And most important, oxidative phosphorylation. Rylation. In other words, the electron transport chain. Electron transport chain. They make ATP. That is the supply energy to the cells. Vacuoles are present. They are fluid-filled sacs that hold several materials. In plants, these vacuoles are very prominent. So, lysosomes are other type of membrane bound. This is a lysosome over here, red color. They contain enzymes to digest material. They are proteolytic enzymes. They break down materials, digest out and break down materials. We have these centrioles. They are tubes found in the centromeres of chromosomes. They are little tubes. They look like this during cell division. All these spindle fibers and all these fibers which are there, they are all originating from centrioles. They help to divide the DNA during cell divisions. From one end to the other end, the centrioles release spindle fibers, and from these spindle fibers, the chromosomes, they move from one side to the other side. The centrioles also form cilia and flagella. The spindle fibers, cilia, flagella, they are all made from centrioles. They form they all originate from the centriole. And so basically, this is the nutshell of the cell theory. So do you people have got any questions? Yes, no, question থাকলে জিজ্ঞেস করো। कोई भी question है, please ask. Ask, type. Yes, no? Okay, I will tell. So since there are no questions, that means there is no questions. Am I audible? Are you people able
Yes,
to hear
sir.
me? Okay,
Yes.
okay.
Yes, sir, you are audible.
Okay. That means there is no questions then. Okay, I'll send you the recording of this, and your attendance has been noted down. I've tracked the attendance. I will hope you people have entered from your own name. Oniket Da. Okay, that's in Bengali. Okay, I will. Okay, I'll remember that. Okay, Vijay Kumar, Tania, Shomen. So anyway, it will be tracked down, and attendance will be given. Day after tomorrow, I will take another class. The day after tomorrow, I will be taking prokaryotic cell structure, or structure of a bacteria. Your exam schedules are out. Do you know them? If they are out, পরীক্ষা কবে হবে? জানো কেউ? হ্যাঁ না বলো কেউ?
4th August.